Okay, good afternoon. I think we'll get started, and it's great to see such a great crowd here. My name is Tim Martinson. I'm the statewide viticulture extension associate with Cornell University, and I'll be the moderator today. I'd like to welcome all of you to our live webinar on Grapevine Red Blotch disease. Most of you are sitting at your computers, but I'd also like to welcome those at Prosser at the Washington State University facility who are viewing this as a group, which was organized by Michelle Moyer up there in Washington State. In addition to being the statewide extension associate, I'm also a member of the National Clean Plant Network Grapes Outreach Committee, and this webinar was organized under the auspices of the NCPN. So I'm very pleased to have my colleague and virologist Marc Fuchs from Cornell, UC Davis Farm Advisor Rhonda Smith, and Deborah Golino, virologist and director of the Foundation Plant Service at UC Davis as speakers today. We expect this webinar to run about an hour and a half with 3 presentations, each separated by a question and answer period. We hope to provide you with our understanding of what we know about red blotch disease and the grapevine red blotch associated virus and what we don't know, and the questions that need to be answered. And I think it's really useful to realize that it's only been since last July that a diagnostic test has been available. And of course, this allowed extensive testing of vines, and it's safe to say that this test has unleashed a fury of activity, interest, and some anxiety throughout the industry. So we'll try and address with that. Just a few words about how to interact. Above all, we want to make this event interactive, have a large audience, but I'd like to encourage everyone to submit questions by typing them into the chat bar. And first of all, you'll want to use this arrow here to drag it to the left to make your thing larger. You can click on this triangle here to minimize the participant list and make the chat bar bigger. This is the chat bar where we'll do a lot of our interaction. You can type in questions at the bottom here, and then if you would, set this to all panelists or all attendees or the most, so that more people can see it, and then you just hit send. Apparently, you can only send comments to the host and the panelists, so we will copy those and paste those in. But please feel free to submit questions at any time. We'll have people monitoring the chat bar while the presentations are going on, and also, excuse me, and also we'll try and address as many questions as we can after the talk. Okay, and we are recording this today, so we will record it and post it on the web, and I'll be happy to send out the link to everybody. At the end of this, you'll be directed to a 5-question exit survey with the opportunity to provide us with feedback. So, in addition to our speakers today, this is going to feature work by Mysore Sudarshana with the USDA ARS at Davis, California. I'm Tim Martinson, and Keith Striegler and Keith Perry are going to also be monitoring the chat lines and be able to respond to questions. And finally, this webinar is a product of the NCPN Grapes Outreach Committee funded by the USDA Farm Bill Section 1021. And with that, I would like to turn it over to Marc Fuchs. So I will make Marc the presenter here, and we'll switch over to Marc's talk. So, Marc?
Thank you very much, Tim. Good afternoon, everyone. Good morning, and greetings to everyone, whether you're in Australia, Chile, Israel, Portugal, India, in the U.S. Pacific Northwest, California, in the Rocky Mountain states, Midwest, in the South, Mid-Atlantic, Northwest. Welcome, and I want to thank you for participating today in making this seminar, webinar a success. First of all, I'd like to make clear that my presentation results from a team effort, and I'm delighted today to just act as one of the spokesmen for Dr. Mysore Sudarshana and Dr. Keith Perry, my colleagues with whom we have done some research on a virus that we believe is associated with red blotch. So today, if Tim can put up eventually, here is my presentation. So my charge today is to talk about Grapevine Red Blotch associated virus and its association with red blotch disease and its presence in vineyards. And let's see, for whatever reason, I cannot move the slides, so bear with me just a sec.
Mark?
Yes, Tim?
Is it showing your outline slide now?
It is showing, yes. No, it is not. It's still on the first one.
Oh, my goodness. Well, I'm able to move it around.
Okay.
So hang on. So it is on your second slide. So is that not what you're seeing, Mark?
No, I'm not seeing it. But okay, that's okay. If you can walk me through, that'll be fine, Tim.
Okay, we're on outline slide.
So outline. I apologize for this technical limitations here. Anyway, today what I'd like to offer is to give you a historical perspective on red blotch and in the broader context of research on virus diseases of grapevines, how this virus was discovered. What do we know about the biology and ecology of this newly discovered virus? And more importantly, what we do not know. Because my ultimate goal today is to summarize not only the state of the art, but also identify gaps in knowledge for which critical research is highly needed so that we are in the best possible position to devise management options and at least make informed recommendations in terms of management. Next slide. So again, as a historical perspective, usually long-lasting efforts, next one, Tim, please, are long-lasting efforts between team of investigators are critical to advance our knowledge and identify and characterize graft transmissible diseases and their related agents. And these efforts very often results or always results from well-thought experimental approaches that are designed based on clearly identified hypotheses, and sometimes a little bit of serendipity might help make major breakthroughs. So as we speak today, to the best of my knowledge, there are 56 viruses that have been identified in grapes. And these 56 viruses are classified into major groups. Viruses that are responsible for degeneration and decline disease, those that are associated with leaf roll. A third group are the viruses that are associated with rugose wood. The fourth group are viruses associated with a fleck disease complex. And there are many other viruses for which an association between the presence of a virus and a disease has not been established yet. Next one. So again, from a historical perspective, there are a number of viruses that belong to these different categories. For example, there are 16 different viruses that are associated with the degeneration decline disease complex. I'd just like to point out here that it took close to 80 years, from the time that some symptoms were described to the time a virus and its potential vector were identified. In the case of leaf roll, it took close to a century for a virus to be associated with leaf roll disease after symptoms were described in the literature. Now, what do we know about cause postulates in terms of these different diseases and viruses? And again, cause postulates is just a way to establish a causal relationship between a microbe, or in our case a virus, and the disease. And cause postulates has been fulfilled exclusively so far with viruses involved in degeneration decline disease complex. And this is because investigators were capable of isolating these viruses, for instance, grapevine fan leaf virus or tomato ring spot virus from infected vines, transfer these viruses to intermediary hosts such as lamb quarters, and carry back these viruses from these herbaceous into grapes, therefore recreating the initial disease. These cost postulates have not been fulfilled for any other virus complexes identified for grapes. However, like in the case of leaf roll, next slide, Tim, please, we have enough confidence that the leaf roll associated viruses that have been described so far are involved in the disease etiology just based on the very high association between the presence of the viruses and the symptoms that they can induce on infected vines. Next slide. So basically, how did we come about the discovery of this newly identified virus? At Cornell University, like at many other research institutions, we have accumulated a number of selections or accessions that show typical or similar symptoms than leaf roll. And all detection assays that were directed to the identification of leaf roll associated viruses failed to identify a potential causal agent. And in addition, all attempts to identify new viruses associated with leaf roll disease or a variant of known viruses have flatly failed. So this was a very puzzling situation for a number of virologists across the US and across the globe. In 2008, next slide please, there were field observations that were made by either vineyards or viticulturists either in California or in New York, where in red-berried Vitis vinifera cultivars, leaf turned red earlier on prior to harvest. Sugar levels were relatively low based on the expectations, and basically ripening was detrimentally affected. And one of these vineyard blocks was a Cabernet Franc block that Justin van de Heuvel, our viticulturist at Cornell University, was managing and called my attention to this particular block. Next slide. So in terms of symptoms, there is a range of symptoms that have been observed so far. And this is just a comparative slide showing a typical leaf roll symptom on a leaf of a Cabernet Franc on the left, and these red blotches on a Cabernet Franc leaf on the right as observed in California. Now, what is also very interesting is the red blotch doesn't show any cupping. Basically, there is no downward rolling, and like for the most commonly observed symptoms on leaf roll affected vines. Another differential characteristic between leaf roll and red blotch is when you look at the primary, secondary, and tertiary veins, you might see some reddening or crimson red colors on red blotch affected vines, a situation that we rarely or do not observe on leaf roll infected vines. And next slide. Again, this reddening of primary, secondary, tertiary veins have different intensities and different magnitudes of discoloration. However, this reddening of primary, secondary, tertiary veins might not be a typical diagnostic symptom for a red blotch, because we have to be mindful that symptoms, and particular virus symptoms, result from the interaction between a virus strain or different virus strains in a given host that in our case consists of a rootstock sign combination in a particular environment. So many factors can influence the intensity and the type of symptoms that one can observe. Next slide, please. And this red blotching and primary vein discoloration and the panoply of this range of symptoms can be observed here, and particularly late in the season around harvest time and beyond, where the crimson red starts to invade the full leaf blade, and you can see on some of these pictures, on some leaves the veins remain green, on others they turn yellow, or the whole leaf blade turns yellow, on which you have this red blotching superimposed. In terms of production, next slide please, if I take my fisheye camera lens and I go under the canopy, I get the picture on the right where yield doesn't seem, at least visually, to be affected by red blotch. And we basically the similar type of production as on a healthy check on the upper left corner, while on the leaf roll affected vine, very often visually speaking, you can see a reduction in yield and obvious symptoms of leaf roll. In New York, next slide please, on Cabernet Franc, these are the type of symptoms that we have seen on field-grown vines. Basically, the similar red blotching at varied intensities, but main veins or secondary veins remain green. Again, illustrating very likely the effect of cultivar, rootstock, virus, environment interactions. How was Grapevine Red Blotch associated virus discovered? Next slide, please. It was basically Dr. Bjorn Krenz, a postdoctoral associate in Dr. Keith Perry's lab, who had a brilliant idea. Next slide, please. And Bjorn's idea was to use the rolling circle amplification methodology to look for the presence of circular DNA molecules. So this methodology is a very simple one, but it states that on the first step, there needs to be a circular DNA present in the host to be surveyed. And then we use an enzyme that is extracted from a bacteriophage that will amplify this template, circular DNA template, exponentially. So basically, you have to imagine that you enter a roundabout, and basically the circular intersection in which traffic flows continuously in one direction. And you continuously go around and amplify this DNA template. And if this DNA template has some restriction sites or some specific markers that can be identified by specific enzymes that will act as molecular scissors, you will be able to cut this amplified DNA template into several distinct pieces, providing a fingerprint for the DNA that was amplified. And this fingerprinting pattern is seen on the right-hand side for Grapevine Red Blotch associated virus. So we initially identified this product. Now the question was, what is the origin of that product? Next slide, please. And this was done by sequencing. In parallel, at Davis, a different approach was used to identify a potential virus that could be associated with red blotch disease. And the approach was to take red blotch disease vine to sequence the entire genome of the diseased vine, and in parallel, do the same with a healthy vine. And if you subtract the data output of the healthy from the disease, the result will be a sequence of a population of microbes that eventually would include the signatures of a potential causal agent. So to make a long story short, next slide, a virus was identified. So the circular DNA that was initially identified shows the typical signatures of a plant virus. And the genetic organization of the virus is consistent with a plant virus. And I'd like to make it very clear that based on these signatures and the information obtained, this virus does not resemble a mycovirus. It does not resemble a virus that infects fungi. It has all the signature trends of a plant virus, including genes that are represented here by colored arrows, either that code for a code protein, code for replicase genes, and likely movement genes. So this being said, the virus that was identified had very unique features, and we try to better understand what type of features and how this virus was related to other known viruses. Next slide. And basically, the sequence of this virus enabled us to classify this Grapevine Red Blotch associated virus within the family Geminiviridae. This is a plant virus family which has quite a wide host range because it infects vegetables, field crops, ornamentals, and papaya. And at the same time, Grapevine Red Blotch associated virus was identified. A very similar virus was identified in Europe in disease citrus. And this virus was named citrus chlorotic dwarf associated virus. Now, what is quite interesting with this virus family, their virus particles are twinned, or Gemini, hence the name of the family here, Geminiviridae. Next slide. I fully acknowledge that the next slide is very busy and might not speak volume to many of us. However, I'd just like to call your attention on the number of arrows that are colored whether in red, pink, or gray on one hand, and those that are colored in dark blue, light blue, or light purplish. And next slide, if I put close to the known viruses within the family Geminiviridae that can be grouped in 4 genera called Mastrovirus, Curtovirus, Topocurovirus, or Bigomovirus, we can see that Grapevine Red Blotch associated virus shares a number of similarities, and without speaking about the general genetic organization, with all these viruses. However, it has distinct features in the numbers of red arrows or blue arrows compared to the known Geminiviridae. So the virus belongs to the family Geminiviridae. However, it has very distinct features. Next slide, please. And if I look at the relationship of Grapevine Red Blotch associated virus with other viruses within that family, what I show here is you have to imagine a tree where the trunk would be to your left-hand side. And all these different lanes represent branches on the scaffold of that tree. And you can see that if I analyze and compare the replicase gene of a Grapevine Red Blotch associated virus with all the virus members of the 4 genera within that family, we can see that Grapevine Red Blotch associated virus is very close to the citrus virus and to members of the genus Mastrovirus. Now, the situation is quite different when I try to put together the same approach and build a tree with a code protein gene. In that case, the citrus virus is related not to Mastrovirus, but to Bigomovirus, and Grapevine Red Blotch associated virus is all by itself on a single branch. So this was quite interesting from a virology standpoint of view. However, I'd like to make clear that given the limited information that is available today on the genetic composition and organization of this virus, it is very difficult, if not impossible, to make any inference on the origin and evolution of this virus. However, the fact that the virus was discovered and that, next slide please, and that the genome was discovered put us in a position to design primers to be used in PCR assays for the detection of this virus. Next slide. At Cornell, in Keith Perry's lab, we use a multiplex PCR approach where primers are designed in the replicase and in the code protein gene, in addition to a grape gene that is used as housekeeping material. And this is just done to increase the confidence we have on the robustness of the assay that we are running. Now, next slide. The next slide is very likely the most important slide of my whole presentation. So I might take a little bit of time to walk you through this slide. And up front, I'd like to give credit to Dr. Sudarshana, who did this fantastic work, trying to find an association between the presence of the virus and symptoms of red blotch. And Sudi surveyed more than 12 vineyards in which symptomatic and asymptomatic vines were present. So, for example, in Vineyard A, he surveyed 38 symptomatic vines, and 32 out of the 38 showed the presence of the red blotch-associated virus. And in 6 out of the 68 symptomless vines in the same vineyard showed the presence of this virus. But if you look at the total at the bottom of this chart, you can see a 96% correlation rate between the presence of Grapevine Red Blotch associated virus and symptomatic vines, and this correlation is only 2.5% in the right-hand column. So 96% put in the broader context of grape virology. And you know, I have been working with grape viruses for close to 30 years. I have never experienced a situation where such a high correlation was found so quickly after the discovery of a new virus. Now, there are 2.5% of the vines that were asymptomatic that showed the presence of the virus. This can be explained by a lag time between the timing of infection and the development of symptoms on some of these vines. Now, based on the assay that was made available thanks to the sequence of the virus that was obtained, next slide please, several infected vines that were either suspect or not for red blotch were surveyed for the presence of Grapevine Red Blotch associated virus. And to my knowledge, this virus, newly discovered virus, has been identified in more than 20 different cultivars and rootstocks in many different viticulture regions in many states. And the ones with red stars indicate the presence of Grapevine Red Blotch associated virus. So, in summary, next slide. What do we know about the biology and ecology of red blotch and Grapevine Red Blotch associated virus? Next one. We know that red blotch is a recently identified disease, that a number of diseased vines have issues with fruit ripening and sugar levels that can be substantially reduced up to 5 bricks. Grapevine Red Blotch associated virus is a newly discovered DNA virus that is closely associated with symptomatic vines showing red blotch symptoms. A PCR assay has been made available for diagnosis. We also know that the virus is graft transmissible because it can be found. It has been found beyond the graft union of a number of grafted vines that were chip budded with buds collected from vines showing red blotch disease. We also have anecdotal evidence that there might be increased incidence over time of the virus in some vineyards in some locations. Next one. So this is what we know. The information, I don't have to emphasize the paucity of the knowledge information that is available on the virus. So what don't we know, and what would be the way to go to fill the gaps in knowledge, and how can research help us get there? Next one. I think one of the major issues to address would be, can we fulfill cause postulates for this virus? Two approaches are possible. Reproduce symptoms on healthy vines. This can be done by grafting with budwood from infected vines onto healthy, monitor the transmission of the virus on the healthy parts of the indicators. Another approach would be to copy or engineer the genome of Grapevine Red Blotch associated virus and move it into Agrobacterium tumefaciens, which is the causal agent of Crohn gall. I should emphasize here the use of modified Agrobacterium strains, because we don't want these strains to cause tumors. We just want them to keep the ability to transfer genetic material into the grape host. And basically, we move the virus genome into Agrobacterium and ask Agrobacterium to deliver it into healthy vines, expecting that the virus might replicate, infect the vines, and express symptoms. Another approach to fulfill cause postulates in the cure symptoms from diseased vines, and the visualization of virus particles might just strengthen this type of analysis. Now, the next question, are the current detection assays robust? We have limited information available on the genetic variability of the virus. Are the primer sets that have been used so far reliable for a broad spectrum identification of all the virus strains and genetic variants that are out there? I think serological assays based on the prediction of antibodies would be also an interesting way to complement the PCR assays, and again to improve the reliability of detection. Next one. I think a third objective would be to have a better understanding on the distribution of the virus within infected vines. What is the seasonal distribution of the virus? How can seed shoot sampling be optimized? What is the latency period for symptoms to develop after infection has occurred? And this issue is very critical for an optimization of detection. Number 4, what is the dispersal of the virus? It is transmitted only by grafting, or is a vector involved? And some preliminary information seems to indicate that a vector might be involved in some vineyards in the dispersal of Grapevine Red Blotch associated virus. We don't know much about the potential interaction of Grapevine Red Blotch associated virus with other viruses. Are these type of relationships synergistic, antagonistic, commensalist? We just do not know. Next one. What about the elimination therapy methodologies that are commonly used? Are they effective against Grapevine Red Blotch associated virus? Is microchute tip culture efficient, or should meristem culture or embryogenic cultures be contemplated for the elimination of these newly discovered virus? We know little on the virulent reaction to infection by Grapevine Red Blotch associated virus. We have some preliminary information on the effect on vine health, but this information is limited to a few cultivars. if we were to go along this line, would we be able to identify tolerant cultivars or tolerant rootstocks? Again, that's a very important line of investigation that merits further efforts. And obviously, what is the situation with the cleanliness of the planting material in relation to the sanitary status of the foundation stocks? And I think these are nice segues for Rhonda and Deborah's presentations later on. But again, all this information is very critical for the scientific community to be in a position to deliver informed information and recommendation for mitigating and the best management of this newly described virus in a newly identified disease. And with no further ado, I'd like to thank you very much for your interest and attention. And I'd like to thank especially Dr. Mysore Sudarshana and Dr. Keith Perry for putting confidence in me, and I hope I didn't misrepresent your respective research and your contributions today. And lastly, I'd like to thank Bjorn and Jeremy, who are spearheading all the research efforts in Dr. Keith Perry's lab at Cornell. Jim Walper, Andrew Walker, Mike Anderson, Michael Sipura, and Justine von de Heuvel, who taught me everything I know and the little I know about red blotch disease, the impact on vine health and on symptomatology. I'd like to thank also Bob Martin, Alan Wade, Judith Moniz, and Alicia Fabritus and Lucy Morton for volunteering information, sharing samples with us, and Tim Martinson and Keith Trigler and NCPN for putting this wonderful seminar and webinar today together today. Thank you.
Okay, thank you, Mark. And I want to apologize for all the technical difficulties we seem to be having. It seems like some people were asked to join a teleconference. There is no teleconference. It's all going through voice over internet protocol. And apparently it's confusing for some of the people who are trying to sign in on that way. So I'm not sure what to do with that, but I want to address just a couple questions that have come in on the chat bar. And one of them is from Patricia Slathers. There are a couple questions. What kind of foliar symptoms do white varietals show? And another one, could red blotch have been mistaken as physiological berry sugar accumulation disorders? And also, what is the mechanism of delayed maturity? So if you can answer any of that, that'd be great.
Let me take a stab at the two first ones. These are very good questions, actually. In white-berried varieties, very often there is a slight chlorosis that has been observed on the foliage. I'm not very familiar with the disease on many white-berried varieties, but that's my personal observation. Maybe Rhonda might chime in a bit later during her presentation to expand a little bit more. But again, this virus being newly identified as well as a disease, I think we have so much more to learn. I'm convinced that the disease was misidentified for a long period of time to either leaf roll or any other potential physiological disorders, although the reddening of the foliage might not have exclusively be related to any physiological disorders, but would have pointed to a graft transmissible agent.
Okay, I have one from David Hubel that I'm going to hold for Rhonda's presentation, I think, or a little later. And that's any indication of vectors likely to spread the virus. If not, when might we know? So I guess we don't know a lot about that yet. Here's one from Michelle Moyer in Washington State. Number one, in commercial vineyards here, we've noticed very stunted shoots on 1 to 2 year old vines that were then identified as having red blotch. Are these early onset symptoms common?
I'm not in a position, that's another very good question, Michelle, thanks for asking it. I'm not in a position to answer this question because I've never observed red blotch in, 2-year in the second year, the second year vineyards. So I'm not in a position to address this question. I'm sorry.
Okay, I got one from Ruben Arias. Can grapevine leaf roll virus and red blotch disease be field surveyed during the same time period as prior to harvest? A good time to survey? If so, in California, what time of the year is best? Fall?
Again, we have limited information as to which is the optimal window during the growing season that would facilitate identification of red blotch. Definitely leaf roll late in the summer, early fall around harvest is an optimal time. This work has not been done for red blotch. It needs to be done. However, based on the limited knowledge we have, I would anticipate, based on the biology we know about the 2 types of diseases, that there should be some overlap. However, more research efforts are really needed to really confirm this type of information.
Okay, one last question here from Annemiek Schilder. Has the disease been found in Europe or other parts of the world?
To my knowledge, yes, the virus has been found in British Columbia, in Canada. I know of ongoing efforts in Europe and Australia and New Zealand, so I wouldn't be surprised based on the distribution, the geographic distribution in the US and the distribution in so many different cultivars that many other countries will identify that virus as well.
Okay, we intend to deal with all the questions that come in, but I think we have to move on here and move on to Rhonda Smith's presentation. And Rhonda, I'm hoping that you can control your own slides, and I'm going to unmute you here. And there you go. So can everybody see Rhonda's slides? Why don't you just go ahead, Rhonda, and thank you, Mark, for your presentation, and keep on putting your questions in. We will record them and record this session for later posting. Thank you.
Thank you, Tim. And just wanted to say that this is my first webinar. It's fascinating. I too had some technical difficulties, so I got to hear part of what Mark said, but not all of it. And also, it does require some serious multitasking to be able to read the responses in the chat bar, questions in the chat bar, and listen at the same time. So we're not ignoring you, and the moderators, the people who are moderating or the monitoring, and as well as moderating the chat bar, they're not ignoring you. And if the speakers seem to be ignoring you, it's because, well, speaking for myself, I only do one thing well, attempt to do one thing well at a time. Okay, that being said, I'm the Viticulture Farm Advisor in Sonoma County, and I was asked by Tim to make a presentation on, as the title says, Field Observations Impact on Vines. I think some of what you're going to hear is going to be a little bit redundant, which is very good, I think. It's sort of nice to be on the same page. As well as a little bit different. There's a different experience in California to some extent, but not dramatically. So the first thing I'm going to be doing is to give you my list of the issues that I would like to go over, how grapevine red blotch virus was identified in California, specifically Napa County, and then, of course, as people started looking harder for it, found it in many other locations. The effect of red blotch virus on disease on vines, as well as how red blotch disease was probably overlooked for some time, some period of years, as well as wrapping up with some grower concerns. So about 2007, not distinctly, not specifically 2007, but pretty close to it. I think Mark mentioned 2008 with his introduction to this issue. But in my experience, there seemed to be an increased incidence of vines with red leaf or disease observed in primarily Napa County, but also I had colleagues point out in my own backyard where there were instances of blocks with red varieties showing symptoms that looked very, very similar to leaf roll disease. It started then. It could have started a lot earlier, but the chatter began. In other words, people started coming together and saying, what's going on here? I see this. These are certified tested wood, certified virus tested wood, or sometimes it wasn't, but I'm seeing an increase on onset of what appears to be leaf roll disease. What's going on? Specifically, over at the UC Davis Oakville station in Napa County, Napa Valley, there were some symptoms specific at that time, or initially observed at that time in Cabernet Sauvignon, by the staff research associate at that time, Jason Benz, who has since retired. He noted that the symptoms of what he thought were just as other growers in Napa Valley and Sonoma County were seeing. They mimic leaf roll, but they weren't really leaf roll-like, not exactly. And also in those blocks that he was addressing, they too tested negative for leaf roll virus. This is a photo that I received from Mike Anderson, who is now the research associate at the Oakville station, since Jason retired a few years ago. This is a photo of Jason holding up a leaf blade that he chose to identify with the terminology of red blotch. And it was in row 1, vine 5 of what Oakville Station refers to as their cover block. A little bit of historical info there, which may or may not be informative, but it definitely was the first photo that I'm aware of something where red blotch was associated with it as being unique from the symptoms that were generically believed to be leaf roll. Well, in 2009, growers as well as USDA virologist Mysore Sudarshana, who I refer to as Sudi because I have a difficulty in saying his name. Several people refer to him as Sudi. Both growers and Sudi submitted samples to various diagnostic labs for virus testing of symptomatic vines of 4 red varieties. The results were negative for leaf roll virus, with one exception. They weren't alone. This had been done repeatedly by many growers, and repeatedly the same answer was being obtained from those labs, and that leaf roll viruses, any one of them was not being detected. So this is an example of a vineyard. It's late fall. There's red leaf symptoms there. Well, it's going to be sort of discerning for a grower to see this increase over time, have it tested, get it tested, and not find any correlation to a known virus. And as grape growers in California as well as other areas are aware of the fact that when you submit samples to a lab and everyone feels and believes that it's definitely a virus as a causal agent, but indeed the lab cannot detect one, it's simply because there was no diagnostic test available for that virus, and which simply leads to continued frustration is going, what's going on? My vines are being affected. In some blocks, not all blocks, ripening issues were occurring that were associated with an increased incidence of vines in these blocks, or these types of vines and blocks. And so there was a lot of unknowns. Busy slide, I apologize for that. It just sort of wraps up the history of what went on in California. In fall of 2010, dormant canes were collected from 3 symptomatic vineyards. So Sudi, as well as UC Davis Foundation Plant Services virologist Maher Alarani, collaborated to process samples of those 3 varieties, Cab Sauv, Cab Franc, and Zinfandel, using next-generation sequencing. Later on, I have a line there, graft transmission studies were initiated by USDA ARS as well as FPS scientists. Those actually began the following year. But anyways, the process began of collecting canes in the fall of 2010. In 2011, a new circular DNA virus, the one that was described by Marc, was discovered in each of those 3 varieties using next-generation sequencing at Davis. And simultaneously, or within that year, researchers at Cornell discovered the identical virus in Cabernet Franc. In July, Marc's team published the results of including the virus genome sequence in the Journal of Virology. And at that time, the virus was known as Grapevine Cabernet Franc associated virus. And then just last October, researchers from all those institutions, plus across virologists from across the world, international conference around the world, basically met at Davis, the 17th meeting of ICGV there. and that's what it is, International Council for the Study of Virus and Virus-like Diseases of the Grapevine, met in Davis, presented their information, and during that conference, just after that conference, the collective decision was the name of the virus was to be modified to Grapevine Red Blotch Associated Virus. So, some of the take-home messages so far is that I just want to emphasize that the ability for commercial labs to detect this virus kicked in after that conference at Davis. So in late October 2012 is when commercial labs got fired up with the primer. They had the primers. I know at least one of the labs did their own sequencing as well as obtained primers from the principal researchers to begin the detection of the samples that they had previously been sent, as well as new ones that were made available to them. And I also want to point out that this disease has been present for some time, for some unknown number of years in vineyards. It's really easy to call it a new disease because the virus is new. I understand that. I will probably slip into that jargon accidentally during this presentation myself. But the disease itself has been present in vines for who knows how long. It's the virus and the classification and characterization of the virus, as well as the primers to detect that virus, is what's new. And I think I'm just going to unplug the phone so it stops ringing. I have no idea how that works. Okay, sorry about that. So, looking at the effect of red blotch disease on vines, as Mark mentioned, at least I think he did, like I said, I got a little distracted with technical difficulties over here. The association of the foliage with red blotch discoloration has been distinctly tied to the presence of that virus. So a virus is not going to cause disease unless there's we recognize a symptom. And a symptom or a collection of symptoms are going to be necessary to basically associate them with the disease. And so not much is going to be said about white varieties, but they can test positive for red blotch virus. I think some of the information in the chat bar back and forth about symptoms on white varieties is critical. We're not really clear on what they are. We have ideas of what they are, and I'm not going to go into our generic assumptions or generic belief. We hope to look at that in a very systematic manner this year, 2013, by establishing trials in vineyards that are positive across a handful of varieties, positive for red blotch virus, potentially, hopefully negative for leaf roll virus, and tracking symptoms in that manner. And as Mark mentioned, virologists are still working on and will soon determine whether the virus causes these or other symptoms, but we're just taking this one step at a time now. So the foliage discoloration has been associated with this virus. And this is going to be a series of photos here, again, specifically to red varieties, associating a white variety with this disease with the canopy still intact last fall. The timeline was a little tight and couldn't really do it that well. But going back in time, November 2011, UC Viticulture Extension Specialist Jim Wolpert, just recently retired, took a series of pictures specifically in Cabernet. I'm going to run through these. As Mark said, there's going to be different symptoms showing up, appearing differently. Symptoms will appear differently in different varieties. And we've seen this tentatively in Merlot versus Cab, just by walking up and down the rows. And as I said later on this year, we hope to characterize the movement of symptoms, the onset of symptom development, and how it differs between varieties, how it differs within a variety, depending on site as well. So looking at the close-up here, the reddening in the veins we've got. We also don't see leaf rolling, excuse me. We don't see leaves rolling, that's better put, but well put, as we do in leaf roll viruses with red blotch leaves. The onset of the reddening sometimes begins at the margins and moves inward, and sometimes it doesn't. That's what my observations are, at least to date. Again, we've got our pinkish veins. Sometimes they're called purple, depending on the grower, depending on the time of year when you're out there looking at them. A lot is not understood about this symptom onset as well as development. The leaf blade does not always become fully involved. It stays blotchy, or sometimes it appears to be. And I believe I heard Mark said that could be because the symptom development or symptom characterization could be because of co-infections with perhaps leaf roll. This is a slide I put in that I took some years ago on a farm call in 2006. It was called out there because this block had ripening issues. And no one really understood why, including myself. We got a backhoe out there, tried to look for problems in the root and the profile, any kind of restricted zones that may have been present. The canopy developed well, but again, this is in July, so this is being tested for red blotch, or it will be this season. But at the time, we all walked away from this going, I really don't know what this is. And I pulled out a file of farm call notes and photos that I have of sites just like this where I really am clueless, and I feel bad about that. And there's more of these than just this slide. And they do involve red blotch symptoms. So moving on, we have you're going to see the word some here quite a bit. I'm sort of the victim of the 5-month window of being able to connect the dots between symptoms that are on vines that are known to test positive by PCR for red blotch virus, as well as the association of that to the bottom line, which in late fall, of course, is the fruit. Some growers, however, were able to act very quickly. They've been documenting these red leaves in their vineyard for some years, and so they were the ones that, in addition to tracking ripening through the course of the end of the growing season last year, the end of the ripening period last year, they were also able to quickly go out and sample vines, or as well as keep lots of vines, fruit from vines that are tested positive for red blotch, separate from those that did not. And they ran their own analysis, and this is their take-home message, is that generally there are lower soluble solids as well as higher titratable acidity, but not always. Not always. There were blocks that I have seen results of from commercial labs, and spoken with growers who have been tracking, as they always do, bricks over several years or TA over several years, and those data are fine. So although the block tested positive, the data associated with fruit maturity on some blocks, unknown number of blocks, are not affected. We don't know the effect on vine growth. So what I mean by vine growth are current season's growth, as well as perhaps, depending on when the vine became infected with the virus, the whole vine size. So, for instance, if a two-bud spur is pruned and then there's a less robust growth out of the basal bud of that two-bud count position, is what is too small to be leaving as a spur? Are you finding the need to find replacement spurs? If vines are hedged consistently after veraison, are they going to be uniform number of amount of canopy being removed, or in a block that has some incidence of red blotch, are vines sometimes making it to the top wire of a catch wire of a vertical shoot position system, or not? So the subtle aspects of vine growth could indeed affect the next step here, which is crop production. Currently, as you saw in Mark's pictures, there doesn't appear to be visually an effect on fruit set. We don't know if cluster number or cluster size, berry number, varies even slightly in diseased vines as compared to vines that are not diseased. Stay tuned for that, something we hope to evaluate this year. Wineries, or well as viticulturists who work for working on estate vineyards for wineries, they were able to process grapes separately. And again, because these are sites that have had the dubious advantage, I should say, of tracking these red leaves or the incidence of these red leaves through various blocks, and they've been able to keep fruit separate. And I know in 2013, this year, there will be more wineries doing the same. And the benefit of that, that collective knowledge is obviously going to help growers and figure out what they're going to be doing about this, to learn if we can mitigate this, and how it can be mitigated in terms of the impacts on vine growth, fruit quality, and potentially wine quality. The next take-home message here is that, well, what it says, we know very, very little about symptoms that can, and the symptoms that can result from this disease. We're very clear on that. One symptom, more is going to be documented, one symptom being the foliage symptom. Other symptoms that are being documented are the ones that growers and wineries have already begun documenting, as well as historical issues of historically how, and I'll get to that in the next slide, of how people perhaps have overlooked this because of the fact that these symptoms have been pretty common. Once we know the symptoms, we'll figure out the range of severity. And this happens to be just a cursory list, a very preliminary list of the things that need to be monitored. I know growers are going to be flagging shoots and tracking things on their own. I just need to reiterate that not all vineyard blocks that test positive for this virus are going to be performing the same, not only in symptomology, foliar symptoms, but also the other effects that I listed on the previous slide. There's going to be a range of symptom severity depending on site as well as management practices. And as Marc said, perhaps there's variants of the virus that could affect disease severity as well. So how is red blotch disease possibly or probably overlooked? Well, you've got similar foliar symptoms as leaf roll disease, which definitely caused people to go back to labs several times trying to find leaf roll disease. And a virus had not been identified. So people were looking for it, people didn't find it. A critical third issue there is that growers have been managing challenging blocks for some time. And they were challenging for various reasons. They didn't have adequate growth, or vine maturity, fruit maturity was delayed for various reasons, including poor soil chemistry, water quality, a lack of water that needed to be applied but couldn't be applied, soils that restricted root growth. There's a ton of reasons why a block is challenging. Some of those had ripening issues, some didn't. And there are also blocks that were not planted with certified virus-tested wood, and some of those are positive for red blotch virus. Some of those are also negative. So people have made a decision in the past to go with uncertified wood. Sometimes they expected to see a virus, sometimes they didn't. But again, not all of those, not by any stretch of the imagination, are positive for red blotch virus. So as a result, people gave excuses to their blocks. They knew they were farming and they had a problem. They did some Herculean efforts in terms of trying to mitigate those problems with fertilization, with soil modification, amendments, et cetera. And many times there was a response, sometimes there wasn't. And also those challenging blocks, and what I mean by challenging blocks are those with ripening issues. The severity of the ripening issue was moderated by year. Over the year, there's not a consistency in some of those blocks, and other blocks there are. So moving on to grower concerns. The first and foremost is a vector moving the virus into vines? Growers in California, as well as outside of the state, have had a large experience with leaf roll disease. And they realize it can cause delay in maturity, and in severe cases, it could actually affect wine quality as well. We've also had the benefit of UC entomologists who have studied intensively in this area. They've been looking at the movement of leaf roll virus into vineyards by mealybugs, the transmission biology of leaf roll viruses by mealybugs, and they've shown that, in 1 year, a vector population can lead to leaf roll disease in previously healthy vines. And all that translates into is that growers here have a great appreciation for viruses that can be potentially vectored by insects. And so as a result, is a vector moving this virus into vines? That's a leading question. 35 minutes ago, I saw it fly by in the chat bar. So it's a question in many, many people's minds. And at this moment, I can say that there's a process to begin to work on that. Researchers are being moved in that direction, are leaping in that direction. And as has been said a few times, this is a new process, and people are getting on board to start the process of the investigation. I said this in a different way prior. Starting in about 2007, growers noticed an increase in the number of plants. Now I'm focusing on those younger plantings, certified stock. Sometimes they were with and without red leaves. Of course, that's a function of if they're white or not. And they didn't have leaf roll virus. 2008 and 2009, I know locally, by locally I mean within California, commercial labs that deal with grapevines had an increased number of samples from, again, as I said before, grape growers saying, this looks like leaf roll, is it? And it wasn't. And they got the no, it wasn't. There was no leaf roll. Growers, as I said before, had a sense, well, something's out there. It's not a virus. They don't have a detection for it, but I'm going to track it. And for some people, they did map it. A small number of growers mapped it, because those of you who have mapped a block for any kind of symptom, it can be challenging. And so I was lucky enough to find a grower who did just that. And this is an example of a block, the incidence of red blotch symptomatic vines, and subset of these, not all of them by any means, have been tested positive by PCR for red blotch disease. Cabernet Sauvignon planted in 2001. There you see the incidence of, and those are rows across the top and vines down the bottom, on an individual vine-by-vine basis. And if you go to the following year, you have an increase of incidence from 46.5 to 51.4. So in some blocks, the incidence appears to be spreading. In other blocks, it appears to exist randomly, but not necessarily be spreading. So this is an evolving knowledge base here. Perhaps those will increase in incidence as well. We're just beginning to collect that data, and we're going to learn and gain information at an incredible rate in the future. So you also have this close association of symptoms, red blotch symptoms, to red blotch virus, and that association was done primarily by Sudi here, by hundreds of samples he collected and which tested positive for by PCR for the virus. And now you have the availability of the PCR primers as of October of last year. A huge number of samples went into commercial labs. As you can imagine, the samples that went in fall came from vines that were symptomatic, and the overwhelming majority of those from the commercial labs tested positive. The ones coming in wintertime, when simple wood is being collected by people who had not marked their vines because, well, they're just either they just heard about the virus, or they're testing a block on their own to see, just for their own information, as what blocks in their ranch may have it or may not. A systematic survey has not been conducted, but I can say that virus has been found in blocks that range from 1 year old to over 20 years old, well over 20 years old. And that vines have tested positive in blocks that did have ripening issues, and vines tested positive in blocks that did not have ripening issues. So it's a little bit over the map, but all over the map, but I think we're going to get there. This is a photo as just what it says. You've got a fourth leaf Cabernet vineyard. Those two vines tested positive for red blotch when I sampled them at that date. 4 days later, the cluster sample in that ranch came back at 22 bricks. So this is a huge concern of growers. You've got a late ripening variety at the end of September, and it's at 22 bricks. And it's delayed. It's not as delayed as it was the previous year. In the same block, which again, different vines tested positive for red blotch the previous year. So this symptomology, characterization of the symptoms, the severity of the symptoms year to year, those are the viticultural aspects of the disease for which we have no clue. So finally, given what is known today, there are some growers making the command decision to postpone replanting a block that was scheduled to be replanted, or else they're going to postpone planting new blocks that they wanted to plant until they understand more about how this virus is spread and how the disease can be managed. And my last slide here, just to reiterate, you have commercial labs having the ability to test for this virus just since October 2012, 5 months ago. So yes, I guess we could call it some respects a new virus. More appropriately, it's a new discovery. And the disease, as I said earlier, has been around for some time. We have no idea how long, but we now have the technology to identify it. Very little is known regarding symptoms, the impact of those symptoms on production, on quality, and if and how the effects of the disease can be mitigated in blocks that are diseased. And ending, I hope, on a happier note, is that work is underway by researchers, including UC, USDA ARS scientists, as well as others, to investigate red blotch disease and learn how this virus spreads. And thank you.
Thank you, Rhonda. And a lot of our questions on the chat bar have been answered by our panelists, but I'm going to still go to a couple of them here. Here's one from New York from Tom Higgins. Would it be your recommendation to immediately remove the vines from the vineyard when they are diagnosed with red blotch virus? Will this virus spread? If not, when might we know?
Good question, Mr. Higgins. I don't know. And when might we know? That's going to be a function of the entomologists looking at movement of the virus, how persistent the virus is in as yet an unidentified vector of the virus. And it's the effects of the transmission. The transmission biology is something that will take a while to unfold of, again, the movement of this virus into a plant with an unknown vector. So the bottom line is, once more, I don't know. I don't know if it's wise to remove rogue, a vine from your vineyard that tests positive for it. It's, as a decision maker, I would suspect it's a function of your tolerance for risk.
Okay. Here's one from Bradley Weeks. I've noticed the symptoms in seedless table grapes as well as their work being done in table grape varieties.
When you say work being done, I'm assuming people tested for the presence of this virus in table grape varieties. That's a possibility. Mr. Weeks, I'm not sure if it has been.
Okay. Here's a question from Ian Bach. Are the asymptomatic positives from the recent survey able to be monitored going forward for insight into the in situ symptom development? And I guess Sudi already answered that. He said, yes, these asymptomatic positives have been tagged to monitor symptom expression. But go ahead.
No, I was just going to say the same thing. Sudi knows exactly which vine he sampled from in hundreds of vines in many, many locations in California.
Okay, here's another one. Have tests been done on rootstocks?
I cannot answer that question. I believe Deborah Golino will be able to answer that question extensively. Okay, well, we're running a little bit behind here, so I want to get to Deborah here, but I'd like to thank you, Rhonda, for that wonderful presentation. Thank you. And I think we'll move on there. And Rhonda, if you can monitor the chat bar and chime in as necessary, that would be great. And I'm going to turn this over now to Deborah Golino. And thank you, Rhonda. And so, Deborah, you're the speaker now, and I'm going to turn it over to your talk. So. Go ahead, Rhonda.
Welcome, everybody. Can you see my first slide, Tim? Okay, so I just heard from my staff in the next room that yes, my slide is up. And so like Rhonda, this is my first webinar. It is wonderful software that Cornell has provided. From my point of view, it makes it very easy for us to do this, and I'm really thankful to Tim for setting this up for us. It's, I think, going to be very useful to us. I know there's a lot of concern about red blotch amongst the grape community in the United States, and I've even gotten phone calls from as far away as Australia on this subject. So I think by having this information posted in detail with this webinar, we will be able to at least let people know that they have good information and that they know what there is to know. We do have unanswered questions, but the research community is going to be working on answering those questions. So first of all, let me introduce myself. I'm the Director of Foundation Plant Services. And Foundation Plant Services is a service center in the College of Agricultural and Environmental Sciences at UC Davis. Its history goes back into the 1950s when the California Department of Food and Agriculture grape certification program was first developed. Foundation Plant Services is has the mission of producing, testing, maintaining elite disease-tested plant propagation material, largely for specialty crop plants. We provide plant importation and quarantine services, virus testing and elimination. We're one of the 3 places in the United States where grapes can be imported, including Cornell and Missouri. And then we coordinate release of UC patented horticultural material as well. And we do a lot of work here in outreach, linking researchers, commercial nurseries, and producers. We have crop programs for grapes, strawberries, trees, roses, sweet potatoes, and pistachios, but our grape program is by far our largest and our most prominent. For grapes, fruit trees, and strawberries, these are formal certification programs run with the oversight of the California Department of Food and Agriculture. So why do you need clean plant material? Well, that may not be a question that we need to ask all of you who are obviously concerned enough to join this webinar, but very often grape growers are not aware of the damage that viruses can do to their vineyards. You see here a scene in Napa Valley where to our left we have healthy green Cabernet that's growing quite well, that is not leaf roll virus infected, and then to the right where we have severe virus symptoms. I still give talks in California after 25 years working in this area where I have growers who have been growing grapevines for many years come up that were unaware until they heard me speak about the effect that viruses can have. Now, the key to planting virus-free material is to plant certified material that's been through a clean plant program like the program at Foundation Plant Services. And I'm going to talk quite a bit about our program, but I would like to acknowledge, as Marc did, that there is a wonderful team of people working on the grapevine viruses. The scientists that attended our ICVG meeting here last fall in Davis work very cooperatively and very closely. And none of the progress we've made in the foundation collection could have been made without the entire team working together. How do we get clean material? This is a very simple overview of the process. New material comes in, we test it for disease, we eliminate the disease using tissue culture or heat treatments. Once a selection has been cleaned free to virus, then it goes into a foundation collection outside but isolated, where it is inspected, professional identification is made that the materials are correct to the cultivar level, and then those vines are registered and sold to commercial nurseries and growers. That makes it sound quite simple. The process is extremely complex. One of my staff referred to it as being like a Rubik's Cube the other day, and I love that analogy. So at Foundation Plant Services, the main diseases we're screening for are the degeneration and decline diseases, the leaf roll viruses, the viruses that cause the rugose wood complex of disease, fleck virus, and then we do test for quite a few minor and rare viruses as well. I need to mention one of the important missions of the program is to provide a safe place for bringing grapevine cultivars to the United States and distributing them free of exotic pests and diseases. All of us know the terrible damage that Pierce's disease can do to grapevines. Some of you will have also heard of plumpox virus, which I believe Marc has worked on quite extensively, a very severe damaging virus in fruit trees that is common in parts of Europe and is not in the United States. A little bit of it got in, smuggled, probably with budwood. That's a very good vehicle for pests and diseases. When we have an important, valuable selection and we want to free it of virus, we use microshoot tip culture. It would seem from some of our data that microshoot tip culture is quite effective also in freeing material of the red blotch virus as well as the viruses where we have more experience. It takes approximately 6 months to get from the 0.5 millimeter or smaller shoot tip that we cut from under a microscope under sterile conditions to a little plant in a test tube like you see here. This is an electron micrograph of a meristem shoot tip, and you can see circled the actual meristematic dome of that growing plant. This is, in fact, a Nicotiana plant, but I think it gives you a real idea of the scale we're talking about here. You can actually see individual cells here in this growing shoot tip. Now, the process for importation and quarantine, or the process for cleanup of materials that already have that are already in the United States but have virus problems, can take many, many years. People are often surprised by the fact that it's truly generational time for new materials to enter the program and then be made available to nurseries and then subsequently made available to grape growers. And so I want you to think about that in terms of this red blotch problem. Creating the nursery screen for red blotch that we are now anxious to do takes a great deal of time. We're very, very fortunate that we have the National Clean Plant Network, which set us on the road to a red blotch-free collection years ago before we even knew what the virus was. So I'm going to talk a little bit about the evolution of our diagnostics at Foundation Plant Services and then take that back to what we are finding in our collection vis-a-vis red blotch. And this kind of evolution has been going on in all of the other modern grape virology labs around the world, but I'm speaking about our local evolution here today. I don't mean to exclude other scientists. We do biological testing for grapevine viruses, and that includes herbaceous testing like you see here, where a little bit of a grapevine is ground up with a mortar and pestle and then smeared on the leaves of plants in which certain viruses will replicate. And I noticed in the chat line, people have talked about the fact that the Nepo viruses, Marc mentioned the Nepo viruses can be detected this way. Everything in our program goes through this herbaceous testing. Biological indexing. This is a kind of test that takes 2 years. We take chip buds of wood from a candidate vine we're going to be testing. We put it into indicators, which will show symptoms after 2 years, and then we grow it out in the field for 2 years. And this is the very, very slow step in our program in terms of grapevine importation and new selections coming into the program. I believe that the Cabernet Franc indicator field index test, you see here a healthy and a virus-infected, leaf roll-infected Cabernet Franc. I believe that the relatively low rate of red blotch in our FPS certified collection is because this test would be positive with red blotch as well, and plants that test positive on the Cabernet Franc are excluded from the program or put into therapy. In the early 1990s, things started to change for virus detection. ELISA tests, which were a simple serological technique that could be run in most laboratories, became available for a number of the grapevine viruses. early on, the problematic virus to test for was always leaf roll. It was very difficult to get Anisura, and very often when we thought leaf roll was in a vine, the ELISA test did not detect it. New molecular techniques came in a little later in the 1990s. Conventional reverse transcriptase PCR came in, and FPS was very much involved with other scientists around the world in developing good PCR assays for the grapevine viruses. Later, led by Dr. Osman in Dr. Adib Rohani's lab here at FPS, we optimized qPCR for grapevine viruses. We can run more samples more quickly, and the qPCR is more sensitive in our hands than conventional PCR, and it's now our routine detection technique at FPS. But still, we have been aware during all these years that there are cases where we see red leaf symptoms where we cannot get a positive laboratory test. And a great deal of our research in the lab all these years from the early 1990s has been trying to get more reliable leaf roll testing. We still consider the Cabernet Franc index our gold standard for testing, but unfortunately that test takes 2 years. Now, I'll explain a little bit more about the National Clean Plant Network in a moment, but the National Clean Plant Network has allowed us to fund Protocol 2010 testing, which is an extensive set of tests well beyond our quarantine regulations and our California state certification regulations for a new collection at FPS. And these are all the tests we do for our Protocol 2010 qualified plants. We are going to be adding the blotch test to that protocol. So what was the next step? The next step was next generation sequencing. This technology is expensive, but it is extremely sensitive, and Marc did a nice job of introducing this technology. I want to give a tribute to Dr. Al-Rohani here in our laboratory. He was the first person to use this technique on a plant pathogen, and it has really revolutionized our ability to find causal agents, viruses, and other causal agents of plant disease. Now, I have some bad news for all of you. I don't think the red blotch virus is going to be the last of these new viruses that we find using next-generation sequencing. However, I believe Rhonda's right. I believe knowing about these viruses may allow us to understand field situations that have stumped us in the past in terms of determining what the causal agent was. The next generation sequencing is almost like a full body scanner for a grapevine. You can find many microbes in the plant, and then figuring out what each of those microbes actually does is a much more difficult process. We got the probe for red blotch very quickly, but figuring out what it actually does in the field, like anything else in viticulture, is going to take us a number of years. So we need to be ready to use this new technology to study diseases of unknown etiology. We need to be able to use it to study diseases caused by new strains of known viruses. And finally, we're going to be using this technology to further characterize existing undiagnosed diseases, things like Syrah decline disease, red blotch disease, graft incompatibility symptoms, leaf roll-like diseases, and grapevine vein clearing diseases of unknown cause. And so I believe we're all going to be hearing about similar stories, similar to the red blotch story, about new viruses quite frequently in the next few years as we discover more microbes using next generation technology. So in our foundation vineyard, we do visual inspections every year, twice a year, and then Dr. Rohani does a very careful inspection of his own in the fall. And for many, many years now, we have seen red leaf symptoms that we could not explain. We've called these red leaf, leaf roll-like, false leaf roll, and now the new term red blotch has been developed. But these have been something that has stumped our laboratory for diagnostics for many years, and as Rhonda says, this is not a new thing. You see here a couple of different samples, 2 vines of Cabernet Franc in our collection. It looks very much like something's wrong with the vine on the left, but all of the laboratory tests are negative. Very often what we do is when we find something like this in the foundation, we go back and do a full woody index. And if that index is positive, the vine comes out of the foundation. So this Ruby Cabernet 03 here, we looked at it with initial assessment, screened for known pathogens, known viruses, with typical laboratory testing, didn't find anything at all. Then Maher performed next-generation sequencing on this vine, and what he found is that there were no viruses known to infect grapevines in this vine, but there were 3 mycofungi viruses, so viruses that infect fungal hosts in this particular Cabernet Franc. In the Ruby Cabernet 03, which also had a red leaf symptom, we found something different. We found grapevine rupestris vein feathering virus, but we did not find the mycoviruses. And I thought about including this slide or not including the slide. I don't want to confuse you, but I do want to get the message across that with this new technology, we're going to have a lot more information about microbes in grapes, and some of those microbes were not going to be clear about what they do and don't do until a few more years of research pass. So how about red blotch in Russell Ranch or the Classic Foundation Vineyard? Well, let's start with the Classic Foundation Vineyard. We haven't tested every vine in it yet. So far, we've tested 1,620 vines. We've got almost 5,000 vines in that collection, so we've still got some work to do. Bud break has already happened in California, and we're not sure whether our testing would be meaningful right now. So until we have a little bit more data about spring and summer testing, I can't tell you when we're going to finish the testing, but we're looking for funding to make sure that every vine in the Classic Foundation is tested. So far, of that 1,620 vines, only 3 vines were positive for red blotch. And that's an extremely low incidence. And so I think our testing and inspecting and optimizing for healthy plants, not just plants that test positive in the lab, has paid off in this red blotch situation. Now it's interesting that the 3 vines that are positive are all different varieties. I'll point out the Tom Cord is a table grape, so we do know that red blotch will go to table grapes. Each of those vines has a sibling vine that was propagated from the same original material that was negative, and that's very hard for us to explain. You can see here the red vine is the one that tested positive, and its neighbors do not test positive for red blotch virus. Now, all these vines are about 20 years old, and this tells us that at least in the Classic Foundation, this virus is not moving quickly. Again, we tested all the neighbors of the Chardonnay that was positive and the Tom Cord that was positive, and none of them had the virus. Now, the National Clean Plant Network is funding that came in the 2008 Farm Bill to help centers like FPS around the country keep modernizing their technology and keep providing better specialty crop material to their nursery clients so that growers could benefit by the additional sustainability of plant material that has been screened for virus. It's been a very successful program, in my opinion, and 4 years ago, the National Grape Network, the Tier 2 network, made the decision to put in a new vineyard at FPS at UC Davis called the Russell Ranch Foundation Vineyard. And I feel very honored that we are able to host that vineyard for the national network, and we're very excited about that vineyard. Every vine in that vineyard has been microshoot tip cultured. Every vine in that vineyard has been through the Protocol 2010 testing and has had negative results. And all of those vines are being Agrobacterium vitis tested for us by Tom Burr's lab at Cornell. The idea behind this is that micro-shoot tip culture gets rid of the viruses you know about, and it also gets rid of the viruses you don't know about. And I think that the fact that this collection was created is a real tribute to the National Clean Plant Network. Not a single vine of the more than 1,100 vines in Russell Ranch was positive for red blotch when we tested it. So our hypothesis that presumptively shoot-tip treating material and putting it in its own isolated virus might avoid future problems with unknown viruses has definitely proven true. And I'm extremely pleased to be able to report that to all of you. Now, here's a little positive red blotch data for you to think about. There is a 25-year-old collection at the National USDA Clonal Germplasm Repository. And here's the results of some of the testing. And I thank Maher and Christine for working through the weekend so that I could have this data to share with you. You can see a very high level, 50% of 20 samples we tested were positive. And look at the diversity of the varieties that are infected. And again, Black Hamburg, I believe, is also a table grape. Thompson Seedless was negative from Mexico, but vines from South Africa, from Spain, from Yugoslavia, Italy, Afghanistan were positive. Now, those vines have been in the United States a lot of years in a mixed planting where we already know there's a lot of virus disease. They could have become infected here in the United States. But it is also possible that they have been carrying that virus since they were imported. And I think our colleagues that attended our meeting in Davis last fall around the world will probably be giving us data on the distribution of these viruses in other parts of the world very, very soon. And so with that, I thank you, and I thank the organizers of this webinar again, and I would be more than glad to answer any questions, Tim.
Okay, thank you, Deborah. And we have several questions here, and I'm going to try and get through a few of them. If Rhonda or Marc want to unmute their microphones and chime in on any of these as well, or offer any final comments, please feel please feel free to do that. Okay, here's a couple from Lucy Garrett to all the, so number one, are vines at the Classic Vineyard Foundation block grafted? And then also, are vines in the germplasm repository grafted?
Yes, vines in the germplasm repository are grafted. Most vines in the Classic Foundation are not. We have nice deep soils, and because of the chance of contaminating our block with rootstocks that might have unknown viruses, we've stayed away from grafting our vines over the years.
Okay, this one is from Karen Keller. She noted very little virus movement in the foundation block. What kind of pest control do they use? Well,
we use a little bit more fungicide than most people do, and we do treat for mealy bugs in the early spring and summer, according to the standard protocols that are recommended by Dr. Kent Dane.
Okay.
We see lots of leaf hoppers in that vineyard, by the way.
Okay. Here's one from Michael Sipiora. Any concern about crown gall moving virus between vines? I would have to answer that probably no.
Yeah, I would answer that no, too.
Okay. Here's one from Patricia Stathers. Could it be possible to trace affected blocks back to the source nursery and then inform growers who had also purchased grapevine material from the same nursery that their vines may be infected with grapevine red blotch associated virus?
I think we're getting at that a little more directly. I think that the nurseries are doing their own self-testing very aggressively now.
Okay, and feel free to chime in on this, any other people. This might be more appropriate for Rhonda, but here's one from Tom Farella. who's wondering if the droughty conditions in California and later harvest might be creating red vine symptoms that are related to vine nutrition and soil effects like pH, leaching, and low fertility in general. I have noticed end vines and lone fence row vines expressing reddening. Most vines don't have red veins or blatant leaf roll symptoms. Yeah. Rhonda, do you want to tackle that one, or Deborah?
Tim, I think you have to empower Rhonda and Marc to join in.
Thank you.
Okay.
Yeah.
You're both unmuted now.
I think I can address Tom's question. I do believe that severity of these symptoms, as well as of red blotch virus, as well as symptoms in vines that appear to be red blotch virus or leaf roll virus, are site dependent and year dependent. So the factors that you pointed out in terms of applied water, other issues associated with the year are definitely going to play a role in symptom expression, and the question then remains verification of what the causal agent of those symptoms. Obviously, if a vine tests negative for red blotch virus as well as leaf roll virus, then it would be potentially a nutritional disorder like, a nutritional deficiency like phosphorus, or a soil moisture availability issue. Okay.
Finally, I think we've dealt with many of these now. And if I've missed some, we'll address them a little later. But here's a last one here from Ian Bach. Have you screened the recent Armstrong Cab Franc trial plants for the virus? I don't know what those are. If not, do you hope to?
We haven't done that yet, but we do hope to. And in fact, we hope to also set up a companion study. Dr. Rohani is working on finding funding for that would also include red blotch in that research vineyard, where we're looking at the effects of leaf roll and other grapevine viruses on multiple rootstocks.
Okay. Well, with that, I think I would like to thank everybody for attending, and I'd like to especially thank Mysore Sudarshana for participating from India, answering a lot of questions, and also Keith Perry and Keith Striegler, who answered a lot of questions in the chat bar. And thanks, of course, to Deborah, Rhonda, and Marc for doing the presentation. As you leave, I hope there will be an exit survey, which is very brief, 5 questions, and you can ask us additional questions. But we basically want to know whether you found this useful or not. And then I'd also like to announce that I am planning on doing an upcoming series that is aimed at more at startup nurseries and extension personnel and consultants, where we hope to discuss, do these at lunchtime and about every 3 weeks or so, and have a kind of an open discussion of nursery pathogens, their biology, and so forth, and practices to prevent their spread. So if you are interested in doing that, you can check a box on that exit survey, or you can email me at tem2@cornell.edu. So finally, I would like to mention again that this is funded by USDA Farm Bill Section 10201 nursery certification funds that were awarded to us as part of this National Clean Plant Network effort. And any reaction you have to this sort of format, I'd really appreciate any comments you might have. And thank you all for attending. I think this has been a very informative event. And if Deborah, Rhonda, or Marc, do you have any final comments?
I'd just like to thank you again, Tim, for setting this up.
Great.
Yeah, me too. Thank you, Tim.
Okay, with that, I think we will sign off. We will leave this open for a while after we all sign out. So if you want to put any more comments or questions in, we'll try to answer them later. And we will also post this webinar, and I'll send you all a follow-up email to indicate where that's located. So thank you all for attending, and we'll talk to you soon again, I hope. Thank you.